Hi everyone, we're starting chapter 16. This is actually looking at gene regulation in bacteria. So we're starting out with the simpler in chapter 16 of looking at bacteria, and in chapter 17 we'll be looking at it in eukaryotes. So as we start looking through this first part we're going to talk about the transcriptional regulation. So hopefully you can think about the different steps of protein synthesis and think about the different places where we could have regulation. The first part will be just looking at transcriptional regulation, and in this we'll talk about the idea of what are called activators and repressors. Hopefully that gives you a hint as to their job. We'll also explain that there are some smaller molecules that can actually affect the function of these activators and repressors, and we'll talk about then how gene regulation can occur in a bacterial cell. So keep in mind that gene regulation is really important at the cellular level, right? It helps decide what will be metabolized. You have to have the enzymes available that can metabolize a given nutrient. It also allows the microbe or the cell to respond to environmental stresses by changing what genes are being expressed and which ones aren't. And then during cell division, obviously we're going to have gene regulation there as well, that it changes during cell division than when a cell is not going through cell division. So keep in mind that this can occur at any point on the pathway to gene expression or protein synthesis. And so we can imagine that it could have control over that transcription period, right? You could have genetic control over the rate of transcription, and even sometimes transcription could begin but maybe be terminated early so that we can have regulation here. Once the mRNA is actually produced, we can actually have some post-transcriptional, so during translation, we can have some sort of regulation. We can have proteins that can bind to the mRNA, which would prevent it from actually being translated. We can have riboswitches that can produce RNA conformation that stops it also from being translated. And then we can have what are called antisense RNA that can bind to the mRNA, and because it's then double-stranded, it won't go through translation. And if we haven't regulated it in transcription translation, we do have the ability at post-translation, once that protein's been made, to actually also regulate that gene expression. We can have feedback inhibition, and that means the product of a given metabolic pathway may inhibit the first enzyme in the pathway. So basically negative feedback, it's like your furnace. If the temperature gets hot enough, it gives feedback to the furnace to shut off. That can happen as well in post-translation. We can also get covalent modifications to the structure of the protein that will actually alter its function. If none of those are actually done to prevent that protein from being expressed, at the end you would have functional proteins. So what's the most common way to regulate gene expression? And you should think about this in terms of energy expended. It makes the most sense to have the most common way to regulate genes at that transcriptional initiation. That means that you're stopping the process as early as you can so that you don't actually expend energy on something that you don't need to. So the most common way is to actually regulate that rate of RNA synthesis, either increase it or decrease it depending on the cell's need. The transcriptional regulation can involve two actions of two main types of regulatory proteins. First of all, we could have negative control, basically the turning off or lowering transcription, and these are called repressors. They repress the actual expression of a particular gene. We can also have positive control, which even though you might have transcription occurring, in this case activators could come in and actually increase that transcription. And that's really important if we need to have an upregulation of a particular gene be expressed. So these activators and repressor proteins can be affected by small effector molecules. So it's not just working on their own, but often they have other smaller molecules that will encourage them in their function. So in most cases, the presence of the small effector molecule will increase transcription. So these molecules are called inducers, meaning that they're increasing or inducing this gene to be expressed. They can function in two ways. They can either bind an activator that allows it to bind the DNA to upregulate that expression, or maybe they bind repressors that prevent it from binding DNA and therefore blocking transcription. So genes that can be induced or encouraged to increase their transcription are termed inducible. And we'll go into some information on inducible when we get into the lac operon later in this chapter. You can also, though, have small effector molecules that can inhibit transcription. So if we want to slow down transcription of a particular gene, these also play a role either with the activators or the repressors. Co-repressors just mean they basically buddy up with the repressor and help it to bind the DNA to stop transcription. We also can get what are called inhibitors that can bind to activators that will prevent them from binding to DNA, and because they're not bound to the DNA, then we don't see an increase in transcription. Genes that are regulated in this manner are called repressible genes, and we will talk about this when we talk about the trip operon in this particular chapter. But to give you a quick overview of it before we get into some specifics, this first two graphs are showing what would happen if we are having an inducible gene, something that could be induced, and that means that depending on the interaction that we could increase transcription. So in the first one, it's showing that there's a repressor protein that is stopping transcription. In this case, in order to have it be induced, we have a inducer that can come along. When it binds with our repressor protein, the conformational change of that protein has it dissociate from the DNA, and at that point then the RNA polymerase can come in and transcribe. So even though there's a repressor present, it can be the transcription can be induced by removing that repressor protein. We also can have an activator protein that actually won't bind the DNA unless it has an inducer bind to it. The binding of the inducer to the activator protein then allows it to bind the DNA, and this will promote transcription. And so in both of these ways, we're seeing the transcription go up. That's because these are both inducible genes. We can have the opposite occur, though, where we have a gene that is what we would call repressible, meaning that it can be repressed. Normally it's turned on, transcription's occurring, but what we can have is a repressor protein along with a co-repressor that, even though this is normally turned on and transcription is occurring, when the co-repressor binds the repressor protein, it will bind that particular start of this gene and it will turn it off. So in this case, that gene is repressible through the use of a co-repressor. We can also have an activator that is attached to DNA that's allowing transcription to occur, and if an inhibitor comes along, it will do a conformational change again to that activator protein, removing it from the DNA and stopping or repressing transcription. In this case, both of these are considered repressible genes, meaning normally they're turned on and that they can be repressed or turned off. So for the checking in, you should think about why a bacteria might want to have an inducible gene and what might cause that gene to be induced. We'll get into this when we start talking about the lac operon. And then think about when a bacteria would want to inhibit transcription. What might be a good candidate for a repressor molecule? And we'll talk about this with the trip operon. But in both of these cases, keep in mind that bacteria are single-celled. They're not dependent on some other cells to produce or provide them with anything, and so they need to conserve their energy by what they need. So you don't really want to be expressing a particular gene if you don't need that product, and you do want to be able to turn on a particular gene if at different times you may need the product of that gene or the protein. But we'll be getting into that in more detail when we cover the lac operon and the trip operon. Again, with any of these lectures, if you have questions, please contact me and I will answer any questions. Thanks.